Welcome back, my biology people. This is the PowerPoint for Chapter 17, covers viruses, which we need to have a little bit of an understanding for to understand how the experimentation process went along to help us discover that DNA was a genetic material, but it's also quite relevant right now given our current situation. The last large pandemic was the Spanish flu pandemic in 1918 that killed 50 million people. That's more people than that died in all of our wars combined. Only about 16 million people died in World War I. And considering the size of the population 102 years ago, that was a large amount of people. And it was very similar in that it was extremely contagious. The military were the first to spread it because they were being shipped off on these really crowded boats. And so if one person on the boat got it, then they all were getting it, and then they were getting off the boat and spreading it. And it was really one of those where there weren't any remedies to save yourself except for just to stay away. And they had very similar recommendations to, have good hygiene, not to go into crowded places, things like that. Don't spit on people. My own great grandmother died of the Spanish flu in 1918. My grandpa was a 1-year-old little boy and was left an orphan. I think his dad died in the war, and his mom died from the flu. So we're going to talk a little bit about how viruses work, and then we'll specifically talk about coronavirus in a few minutes. So viruses come in all shapes and sizes. You can see here, these are all images of different viruses, and the names are off to the side. Things that they cause. They infect different types of organisms. So there are viruses that infect humans, obviously. There are viruses that infect plants. There are viruses that infect bacteria. So we're going to talk about how those viruses work. Many people call viruses germs. Even our own president said that just the other day on the news, suggesting that this was a bacteria of sorts, or like a bacteria. But this really isn't correct. Viruses really are considered non-living. They don't fulfill all the requirements that are needed to be considered living. They don't do their own metabolism, so they don't make their own ATP. They don't respond to stimuli. They don't reproduce on their own. But what's tricky about them is that they do have DNA, and their DNA contain genes that code for proteins, and they are subject to natural selection, so they can change over time. So if you ever wondered why you have to get a new flu shot every year, is because the flu virus actually mutates, and so we have to make sure that we are taking into consideration what the mutated flu version is going to maybe look like in the future, so that's why we make a new vaccine every year. Okay, so viruses are way smaller than cells. You can see here this pink box off to the right here is the size of maybe a bacteria, maybe an E. coli. And these viruses are what fit inside. You can see how much smaller they are. So it's the polio virus here. Here's the flu. Here's herpes, for example. So they're way smaller than a bacteria. The virus's structure is pretty similar from virus to virus. There's a couple of variations, but for the most part, all viruses have some sort of genetic material, either DNA or RNA, and that genetic material is encapsulated within a coat made of protein. So it's called a capsid. All right, so you can kind of see here, this purple here is our capsid proteins, and then surrounding the DNA that's on the inside. Here's another capsid protein surrounding the DNA that's on the inside. Here's one that's an RNA, and the capsid is this black kind of weird shaped thing, okay? Some viruses also have an envelope, like this one does here, which is made of phospholipids. And just like the phospholipids around cells, those phospholipids can have proteins injected in them as well, just like we saw with our membrane proteins. So looking at it here, what's the other really big piece of evidence suggesting that they're not living? They have DNA that has ability to make proteins. They can have a membrane. What are they really missing? They probably had a fluidy material on the inside. They're missing ribosomes, right? So they, even though they have the DNA, they don't have the machinery required to make the proteins from their DNA. That's why they have to infect another cell. All right, so this is just a general picture of what you might see. Your genetic material on the inside. Like I said, it can be DNA, it can be RNA, it can be single-stranded, it can be double-stranded, it can be circular, it can be linear. Then you're going to have proteins that surround that. And then maybe you'll have a membrane, and you might have additional proteins sticking out of that membrane. They call the membrane an envelope. This is actually the coronavirus structure. It is a single-stranded RNA virus. So this is the RNA here. You can see that the RNA is within a capsid of proteins, and then it's bound by a membrane, which has then proteins that stick out of that membrane. These are some other viruses. So this is called the T4 bacteriophage, which we're going to talk a little bit more about this one when we get to our DNA chapter. This one infects E. coli. This is a virus structure that infects plants, tobacco, for example. And then this is the influenza virus, which looks a little bit more like what we saw with the coronavirus. All right, so here's a good thought question. Viruses can't be treated with antibiotics. One of the things that antibiotics are useful for is they block cell wall synthesis. What other part do viruses not have that antibiotics often target? That's right. Antibiotics often target translation, so they also are lacking the ribosomes. All right. And so in order to treat viruses, we typically use vaccines. All right. And if we're going to make a vaccine, which is something we'll talk about in a minute, that stimulates your immune system, what part of the virus do you think you would want your immune system to recognize to destroy it? Yeah, you're right. Probably these proteins that stick off to the outside so that our body can recognize it and get rid of it immediately. Now, when we're looking at viruses, like I showed you in these previous pictures, there's so many different kinds, and so we can classify them in a variety of different ways. We can look at their shape or their size. We can look at what organism they infect. One of the most common ways to classify viruses is by their DNA. Are they a DNA virus? Or are they an RNA virus? And then you can get even more complicated. Is the DNA single-stranded or double-stranded? Is the RNA single-stranded or double-stranded? Is it linear? So here are just some other pictures of various viruses, and you can see how very different they look from one kind to another. Okay, so what about the hosts? So we know they're non-living. We know they can't reproduce on their own. So they kind of live what's called a borrowed life, because they rely on their host to do the work, to make proteins for them. So they do the central dogma using the host's central dogma machinery, and to divide them. And they do that using the host's division machinery. Okay? So we know that viruses can infect all different kinds of organisms. Sometimes they can jump. So you've heard about things jumping from birds to humans or from pigs to humans, avian flu, swine flu, right? They're thinking that this coronavirus actually probably came from maybe bats, is what I think they were thinking. So how does this work? Well, it turns out the virus determines its host by its ability to recognize proteins on the host cell surface. So if you look here, here's my virus right next to the letter B, and sticking out of that virus are a bunch of proteins. So we just saw that in those images. And so the virus is kind of bouncing along cells waiting to bind. And if the right receptor proteins on the cell are bound by the virus, then the cell will actually the virus basically is capable of docking, and the cell can actually get infected with that virus. All right, so let's do a little concept check. Which feature below is not found in all viruses? Hopefully you're saying envelope. That's right. Only some have that outer phospholipid bilayer, but they all have genetic material and they all have a capsid made. All right, let's talk about how viruses actually infect. All right. And when they infect, they get into the cell, and then their job really is, once they're in there, is to kind of commandeer the cell, take over, and dictate that the cell make new viruses while it's in there. So the 5 steps that have to happen is that first the virus has to attach. That has to do with recognition between viral protein and receptor protein on the cell. Then the virus has to penetrate. And that differs from virus to virus. That either means put their genetic material on the inside of the cell, or sometimes the entire virus gets engulfed by the cell. Then the virus has to go through the synthesis phase, which means it's going to take the genetic material that it has and undergo central dogma to make the viral proteins that it needs. Once it makes the viral proteins, it also might go through the process of copying its genetic material, and then it will go through assembly. Well, it will put those viruses back together, and then it will cause the cell to release them. That was an example of how when the cell engulfs it, this is what happens when the cell just injects it. So the virus lands on the cell, recognizes. Once it's got a stable binding place, then it will kind of penetrate through the cell membrane, and then it kind of squirts its DNA through that penetration. All right, so like I said, there's 2 kinds of pathways. You can have the endocytic route, which uses endocytosis to bring in the virus, or there's the non-endocytic route, which is more like penetration, where it binds and then it squirts its information into the cell. So what do you think the source of energy and raw materials? So raw materials probably need nucleotides, DNA and RNA, and amino acids. Where do you think the viruses get those things? That's right, from the host cell. Now, viruses can immediately kill cells, or they can do it in a delayed fashion. All right. When they kill the cells immediately, they're considered lytic. So if you think back about the word we used, lysis means to break. We talked about hydrolytic enzymes are ones that break up molecules. So the lytic infection cycle of a virus is the one that's going to immediately break open the cell. So in a lytic infection, the virus will attach, penetrate, synthesize, assemble, and then release a whole new bunch of new viruses all at once. During that process, the cell will die. There has been talk of using something called phage therapy. So the viruses that look like this that attack bacteria are called bacteriophages. And you can imagine if you could have them target cells that are mutated, cancer cells, for example, then they could bind and cause those cells to die. But if those were the only cells they target, you would be getting rid of the cells that you didn't want. So it's an interesting way of fighting against cells you don't want in your body. All right, so here's the virus. It joins, it squirts in its own DNA, it causes this host DNA to break up. This DNA gets replicated, it gets transcribed, it gets translated, new proteins assemble into new viral particles, and then eventually those viral particles burst out of the cell and the cell dies. All right, same thing here. They burst out of the cell and the cell dies. So one viral particle infecting one cell can end up releasing hundreds of viral particles when they kill that cell. So let's look at COVID-19's lytic cell cycle. So it will recognize the cell surface, recognizes a protein receptor on the cell surface called ACE2. And I do want you to watch a video about this. It then injects its RNA into the cell. That RNA is then used for translation to be made into some proteins. And then it makes a replication transcription complex. So that takes that RNA and it goes, some of it goes through transcription. Those RNAs get cut into different pieces to be made into different proteins. We make all of the different proteins that are necessary. It looks like, even you can see here, we're going through the secretory pathway to make those proteins. And then those proteins are then assembled into viral particles that then release from the cell using exocytosis. In addition to being lytic, viruses can also go through what's called lysogenic, where basically the virus can kind of hide away and not be infectious immediately. A lysogenic infection happens when the DNA that gets injected into the cell, instead of cutting up the host DNA, actually inserts itself into the host DNA and then just lives as part of the host cell DNA for quite a while until there is some stimulus that stimulates it to pop out. So here's the virus infecting, it squirts in its DNA. The DNA then incorporates it. It undergoes many rounds of cell replication, passing along that viral DNA every time. And then at some point, there's some sort of stress or stimulus that triggers the virus to come back out of the host DNA, and then it will go through the lytic cycle and make bunches and bunches and bunches of new virus. Okay, so one example of a lysogenic virus that kind of sits around for a long time is the herpes virus. So if you've ever had a cold sore is caused by herpes. And you only really get them when your body is under some sort of stress. So maybe when you're sick or maybe when you're stressed, all of a sudden you feel that right on your lip somewhere there's a sore, right? And so that's causing those cells to release some of that virus, and you're getting a little viral infection right there where the cold sore is. Other viruses can live even longer. So HIV, for example, can live inside the cells for quite a long time. So can HBV. So you might be exposed to these things, but you don't really have much of a reaction until much later after the exposure. Now, how do we treat viruses? Well, we have to rely on drugs and vaccines. We can't use antibiotics. So antiviral drugs are ones that we can use that interfere with processes or proteins that are unique to viruses. It's hard to find a drug that's virus specific that's not also going to interfere with the host cell. Vaccinations, on the other hand, are a way for us to utilize the virus as is to stimulate an immune response that is not dangerous, so that later when the real virus comes around, our body recognizes it. So what a vaccination does is it basically provides pieces or dead virus, presents those to our immune cells. Our immune cells say, hey, that's not right. We don't want this here. They generate a primary immune response, which maybe give you a little fever, maybe make you feel achy after you get the vaccination. But when you are exposed to the actual virus as a whole, your body's already prepared, it's been through that primary immune response, that it will be a secondary immune response, which is far greater, and it's going to get rid of the virus before you even know you were exposed. Vaccinations have really eliminated a lot of deadly diseases in our world. So, for example, this is measles. So many people died from measles. Measles, very contagious, um, and spreads very easily. When the vaccine was introduced, the numbers of measles just dropped almost down to nothing. But in order to maintain that, you have to keep people in your society vaccinated, which is why we still vaccinate children when they're born for measles. Polio is another example of a virus that really was reduced to nothing after the vaccination was created. So, a couple of reminders: the polio vaccine was actually developed in HeLa cells. Um, and you can see here how many people had polio, and then the vaccine was introduced, and after that, nobody got polio. Whereas people that get the flu shot, look how many people get the flu 0 times if they get annual flu shots. Some of those people get it once, and it drops down drastically after that. And these are statistically significant data. The different types of vaccines that are available are interesting. So you could potentially do a live vaccine where you just inoculate someone with a very low amount of the virus with the hopes that, one, our immune system can get rid of it quick enough, but build that primary immune response at the same time. We also have viruses that vaccines that are made from live, what are called attenuated virus. So what that means is, I told you that viruses have very specific host cells. So what they do is, to make an attenuated vaccine, they take the virus that maybe, let's just say it's a virus that infects humans, and they inject it into a chicken. Well, the chicken doesn't get sick from that virus. So then they bleed the chicken and get the virus back out of it, and then they inject it in the chicken again, and then they bleed it. And they keep doing that, which basically is going to basically force a situation of natural selection to happen. So eventually you're going to find that the virus mutates enough that it is one that actually is not the same virus that infected humans. You take the virus out of that, it's probably no longer able to infect humans because it's changed so much. And so you put that virus into humans with the hopes that it stimulates an immune response. Those are oftentimes administered up your nose. And then you have the inactivated vaccines, where you basically are just taking the virus, killing it, chopping it up into pieces, and then giving it to the person. All right, those are the three most common. That's what we typically get when we get our flu shots and our polios and our rabies vaccines and things like that.